coming along today. I'm going to use some of their empty slots to record some of my residency talks so we can keep getting those done. So it's nice to see some residents here. And today I'm going to start on the first part of a two-part lecture series on immunisation. So today I'm going to talk about some of the background, some of the science, some of the boring bits, and then next week I'll talk about all the clinical vaccination things, so the diseases, the vaccines themselves, and the programs around them. So, see how we go. So today's learning objectives are to describe the principles of vaccination and immunisation and how they're different, identify the standard vaccinations recommended in Australia, and there's some information about pharmacists in vaccination programs. So what is the difference between vaccination and immunisation? Vaccination is the act of physically getting the vaccine. It's the injection itself. Immunisation is the process of receiving a vaccine and then becoming immune to the disease. So immunisation is the process of the immune response to the vaccination itself. In life, most people use these terms interchangeably, but their meanings aren't the same. And immunisation is a term used most commonly in the community and the most correct term because it's referring to the overall process of the immune response. And the vaccination is just a pinpoint snapshot of that process. So some background to immunity. There are a few concepts of immunity we need to understand why vaccination is such an important part of public health. Immunity comes in two ways. It can be either innate or acquired. So an innate immunity factor is something that you're born with. You're naturally able to have your immune system do. An acquired immunity is something you develop in response to an antigen. You can acquire immunity in either a natural or an artificial way, and in both states you can get either active or passive immunisation. So natural immunity is when you are exposed to an antigen in its natural environment. So you get an infection, or antibodies cross the placenta in a passive way, providing immunity that was produced by the mum to the baby. You can develop immunity artificially is when you are exposed to an antigen intentionally. So you haven't picked it up in its natural state or form or place. You've been given it artificially either as a vaccination, which produces an active immune response to develop an active immunity to something, or passively, which is serotherapy. So when we administer patients immunoglobulins in response to a disease, your body's not doing anything there, we're just passively giving you the immunity to it. Does that make sense to everybody? Active immunity is what we're aiming for, and that's what's most important because it's long-lasting and it's able to be reactivated following re-exposure to an antigen, whereas passive immunity only lasts for the lifetime of that antibody itself. So it's a transient state as opposed to something that your body can call on at any time to protect itself from an infection. So which diseases do we vaccinate against? They're usually diseases that are less complex with their own structure and form, so viruses as opposed to bacteria. They're diseases that aren't evolving rapidly because something that's evolving and changing quickly is harder to develop the immune response to because it's going to be different every time you're exposed to. We aim to vaccinate against diseases which are causing serious health problems. While we'd all love to have a vaccination for the common cold, it doesn't actually cause a lot of death and destruction around the place. Whereas preventing measles, which causes lots of really serious health problems, offers massive benefits to our society. Also, vaccines are developed for diseases that are highly contagious. So the faster something spreads, the less effective physical barriers are going to be. Isolation, coughing masks, all those sort of things, because it's already spread faster than you can tell. Whereas a vaccine, where everybody is developing their immunity up following re-exposure, works a lot faster than that. So highly contagious diseases using a vaccine to create herd immunity stops the spread a lot more effectively than anything else. So they're all ideal targets for vaccination. So a brief history of vaccination. Vaccinations have always been known. So there's way back in history there's cases of people exposing themselves to certain diseases to prevent other diseases or have a milder form of a disease. In modern medicine, the first true vaccination was Edward Jenner, who identified that milkmaids who got exposed to cowpox didn't get smallpox, so then went around giving everybody cowpox, which looks kind of the same, but was probably better for the people who got it through the immunisation stents than the people who were touching the cows all the time. And then we have Louis Pasteur, who was around in the late 1800s in France. He developed vaccines to cholera, rabies and anthrax and really started vaccination science off on its current growth and importance in public health. And then we had Sabin and Salk, who in the 1950s independently developed different polio vaccines, but were in a massive move forward in the 50s with polio vaccination. This was the first big public health campaign for vaccines. It was done in the United States as part of the March of Dimes, where everybody donated their 10-cent pieces in order to crowd fund, essentially, a vaccine program. And it was a public health program where everybody could queue up and get vaccinated, and had a huge uptake. And it was the first program like that of its kind. The WHO in 1979 declared smallpox eradicated. That is entirely related to its vaccine program, and it's the greatest success story of public health vaccination programs. We completely removed a disease which caused large-scale death and morbidity from the population through targeted vaccinations. I'm very pro-vaccine. I probably should have declared that at the beginning. And if anyone's not, we can talk about that later. Okay. So smallpox, we found it in Egyptian mummies. It's been around for a long time. Millions of people have been killed in smallpox outbreaks throughout history. It was a very, very serious disease. You see pictures. Has anyone seen pictures of it? I have some in the talk. Basically, people's skin just kind of blistered up, and then they died of bacterial superinfection because all the pustules got infected, and it was pre-antibiotic era. So lots and lots of people died. It was wiped out entire populations in South America and things like that. So very important. Mass vaccination campaigns were run by the WHO across the world, working from location to location across the 60s and 70s, and it took until 1979 to prove that there had been no diseases, no smallpox passed along. And the reason smallpox was a great candidate for this kind of vaccine program was the fact that it doesn't live in anybody else. So it can't live in an animal, it doesn't live in the environment, so it can only be spread person to person. And the vaccine itself was very effective. So each vaccine has different levels of effectiveness, and smallpox is particularly an effective vaccine. It was also quite easy to vaccinate people because they only needed one dose. So you didn't rely on people coming back and compliance and all these things. So it takes a lot of different factors to make these programs a success. Similar programs have been going on for as long as I can remember for polio, and it's been less of a success, and that's partly because polio does live in the environment. It lives in the waterways, so the vaccine, no matter how high you get your vaccine penetration up, there is always going to be that circulating. So anyone who hasn't been vaccinated can still reacquire the disease. So other examples where these large-scale programs and development of vaccines have been not so successful. Everyone's heard of the hope of a HIV vaccine, yeah? It's never been successful because the virus itself is changing too fast. So every time you develop an antigen plan for HIV, there's another rapid evolutionary step in that virus. Oral polio vaccines are not as successful as injected polio vaccines, and it's because it's an attenuated live virus as opposed to an inactivated virus, which means there can be vaccine-induced polio cases, which has led to some of the fear around vaccines in society today. It's not used in Western society, but it is still used in India and Africa for vaccine campaigns, and that is causing some issues there. The pertussis vaccine, which is one I hope everyone here is pretty familiar with. It's a big public health program across Australia at the moment to get more people vaccinated against whooping cough. Back in the 80s, everyone got vaccinated with a whole cell vaccine, which was very, very effective, but caused lots of adverse effects, febrile convulsions, quite serious local reactions. So they developed a vaccine which caused less adverse effects, but it's also been less effective. So we need to do more vaccines, give more regular revaccinations, and even then, our best hope with the acellular pertussis vaccine is about an 80% success rate for vaccinations, which is why it's so important to have as many people vaccinated as possible. So it's a bit of a trade-off there between the adverse effects and the effectiveness. And Panvax. Anyone remember 2009? We finally got a H1N1 influenza pandemic vaccine, but it came too late. The disease was already in its natural decline, and there wasn't a lot of point to that vaccination program because it took us a long time to develop it. I mean, you know, all credit to those scientists. Vaccines are hard, and they did get there. It was just a bit too slow. So lots of things affecting the success of these programs. So now we get to talk about some science, the different types of vaccines and why different vaccines are different. So most common, I'll just go through the most common types of vaccines. Live attenuated vaccines use a weakened but still alive and possibly infectious form of an organism, both bacteria and viruses, most likely viruses. The only live attenuated bacterial vaccine is the BCG. for tuberculosis. These vaccines, use the organism. They've been changed in some way, either exposed to a chemical or a different temperature outside of its sort of happy reproductive range to make sure it doesn't reproduce readily. It's designed to produce the immune response by activating your antibody system and your immune system in your body and then die and not be passed on. But this isn't always 100% successful, and this is why we can't give these vaccines to immunocompromised patients, because there is the chance of activating the infection. Live attenuated viruses and bacterial vaccines are very effective at producing lifelong immunity. So these vaccines can produce immunity with 1 or 2 doses. They don't usually require boosters. They don't require complex courses. They're very quick and easy ones to do. So very effective, have some risk, which is why we don't use live attenuated for everything. Inactivated viruses are a killed virus, usually with formaldehyde. The virus is recognised by the immune system because it's structurally intact, which means as antigens are presented on the surface, which can cause the antibody response and the immune system effectiveness there. They're not as effective as producing the immune response because they are dead, there is some degradation, and we are relying a lot on their structural integrity. So several doses might be needed. They might need 3 or 4 doses to make it through the course for these. With these, not all people will produce an immune response, so they're not as effective as a live attenuated viruses, which is much closer to a natural exposure to a disease. They can be given to immunocompromised people, pregnant women, just about everybody. So it's about looking at the risk benefits again with picking this sort of vaccine formulation. Some vaccines are based just on specific parts of viruses and bacteria. For example, meningococcal, we use the polysaccharide coat, which has the antigens which cause the immune activation on it. We take that off the bacteria and we develop that into the vaccine as opposed to using the whole bacteria itself. So polysaccharide coats are the most common bacterial part used in a vaccine. We also can use some different proteins and things like that. So it's about identifying which part of the organism induces the immune response and separating that out from the part that makes it infectious. Can be given to immunocompromised people. Again, not everybody will produce a response because it's further and further away from a natural exposure to an antigen. And these antigens may have to be combined with something to stimulate the immune system. So we call them an adjuvant. We attach the polysaccharide to something that will help present it to the immune system in a way that makes it more likely to be identified, picked up, and the antibody response produced. So it can be attached to another protein that makes it a bit bigger, makes it a bit more visible, or aluminium is our most common vaccination adjuvant that we use. A toxoid vaccine is different again. So this is about a bacteria or virus that produces a toxin. So the vaccine can be developed using the toxin to induce antitoxins. And the most common one we do for this one is tetanus. So the immune system doesn't recognise the tetanus bacteria, it recognises the tetanus toxin and fights it rather than the organism itself. And these are really good for diseases where the toxin causes the symptoms, tetanus, diphtheria. They're also good for slow-growing organisms for whom the immune response might take a long time at a secondary exposure following the vaccination to identify the organism itself. For example, tetanus is very slow-growing, usually not in a vascularized area, but the toxin will get into your system quickly, so their immune response happens to the toxin, and it's happening a lot faster. So there are lots of different ways to stimulate the immune system in order to get an active immune response out in an artificial way. Everyone cool? All right. So vaccines work by presenting the body with a less or non-infectious antigen to allow production of long-lasting antibodies. So when you are exposed to an antigen initially, you have a primary immune response. That's usually quite small. It can be variably successful in attacking or fighting off the disease, but usually if the disease doesn't kill you, you get better and you keep moving on. What happens then is the next time a secondary exposure to that antigen occurs and you've got a secondary immune response, the response happens faster, there we go, and then it's also larger. So because your body recognises the antigen, it is able to remember what antibodies it produced last time and then produce them quicker. So the first time it's got to really think about it, it's got to develop the antibodies for itself for the first time, but the second time it's being stored in your memory cells in your immune system and they can be released quickly and it can remember how to make them quickly. I like to think of it as like riding a bike. Once you learn it for the first time, you never forget. So your immune system works exactly the same way, and that's why vaccines are so effective. So that brings us to herd immunity. Has everyone heard of this concept? Thanks, Val. Very punny. So herd immunity is the most important thing we do with vaccines. Vaccines is not something, while they protect ourselves, vaccines aren't necessarily something we get for ourselves. They're something we get for everybody. So vaccines are most effective if we have enough vaccination in the community to prevent a disease spreading. So if our susceptible people are here in yellow and are infected, and this is our infected person, this infected person cannot get to any yellow people, yeah? He's surrounded by black people who are our immune people. So by surrounding our people who can't be vaccinated or don't produce an immune response, so people with cancer, people with malfunctioning immune systems, by surrounding them with other people who are immune, we can stop the spread of a disease through our community. So each individual disease has a different level of vaccination that's required to provide herd immunity. So for diseases like measles that are spread very, very quickly, it needs almost the whole population to be vaccinated. What happens down here, if people have stopped getting vaccinations and they're not immune, our yellow people are easily infected by our infectious person because there's no people who are immune to protect them. There's no wall around them. So everybody who comes in contact with our infected person becomes infected and then spreads it to some more people. So herd immunity is about keeping the level of vaccination at the appropriate level to stop transmission of a disease. And that's why it's an important public health concept, and it's an important thing that everyone does as a community as opposed to an individualised thing. Because there are always people who aren't going to produce an immune response, and there's always going to be people with cancer or other conditions that prevent vaccination. Yeah. Okay, vaccines can be given in lots and lots of ways. IM injections are the really painful ones that everybody really does not enjoy. So tetanus, diphtheria, pertussis, all the boostrixes, Hep A, Hep B, Hib, Haemophilus influenzae, HPV, pneumococcal and typhoid given IM, subcut, so less painful, usually a smaller volume, IPV, varicella, Q fever, Japanese encephalitis and MMR, intradermal, so not given as deeply into the skin, influenza, BCG, and oral, rotavirus, typhoid, cholera and oral polio. So lots of different ways to give vaccines. Most of them are given IM, and that's mainly because that's the easiest way to get the antigen into the immune system. Nothing is ever given IV, which is a common misconception about vaccinations, but you want them to be deep enough that the immune cells that you need to pick up the antigen are able to do it. So usually that's in the IM space where there's some vascularization and some exposure to the white blood cells. When you administer vaccines, and this is a pretty common question we'll generally be asked as pharmacists, is can these vaccines be given together? And most of them can. So killed vaccines can be given with other killed vaccines or other live vaccines, and there's no need to separate them if not given together. But live vaccines need to be separated by 4 weeks if not given together. So you can give them all at once, or you separate them by 4 weeks. And that's about the immune response related to those organisms. Because if there's a highly active immune response to a vaccination, say 2 weeks in, and you give a second live vaccine, the immune response is already distracted, so it can't pick up the antigen as well, so it's less effective. So the immune system, like everyone, or a computer, can only do so many things at once. So it's about timing that to get the right response. In children, we give vaccines into different sites to adults. So in adults, it's usually given into the deltoid. In children, it's given into the anterolateral thigh. And that's mainly because it's an easy way to hold a child. You hold them in a hug hold so they can go into the side of their leg. Combination vaccines offer a lot of advantages. Less needles for more vaccination wins. And it's easier to be compliant if you only have to remember one thing.
Sorry.
Yeah.
When you say live vaccine separated by 4 weeks, you're talking about subsequent doses of the same vaccine?
Or any other live vaccines.
Or
So any live vaccine needs to be either given at once or separated by 4 weeks. So either subsequent doses or any other vaccines if you're trying to catch someone up or anything like that.
So if you've got a live vaccine, you can give killed vaccines the following week?
No, you would recommend waiting for 4 weeks.
Wait for 4 weeks
Yeah.
regardless. Okay, thanks.
So a common question you'll be asked particularly if you work in peds, is it safe for my child to have so many vaccines at once? So the Boostrix has got 5 or 6 vaccines in it, depending on which one you get. And these are safe, and they've shown a lot of benefits. So vaccines have less antigen in them than a child would be exposed to when playing, particularly if they're in a McDonald's playground, because they're gross. So vaccines are very safe, and they only have a very small amount of actual infectious or antigenic tissue in it. So the immune response your child's getting from a vaccine is similar to what they come across, and in fact probably lower in day-to-day life. Combination vaccines have been carefully studied to ensure that each vaccine component still has an equitable response than it would be if they were given every vaccine individually. Less needles plus less visits equals improved compliance, and if anyone's ever tried to manage the paediatric vaccination schedule, if you had to give all of those vaccines separately in separate visits, you might as well just move into your GP's car park. And what happens is that people just forget because it's complicated. Whereas if you can rock up, get one needle, come back in 6 weeks, get another needle, and you've done most of your childhood vaccines, it makes life a lot easier. And as I said, vaccination's for the community, so it's important. Adverse effects to vaccines. Immediately post-vaccination, minutes to hours, it hurts. Yeah? Big needle, goes in your arm, hurts. Days to weeks after, you can have a fever, an injection site reaction, you can feel a little bit of malaise, it feels a little bit like a flu. That's your immune response happening. It is not a flu or anything like that, it's part of your immune response. Fever is part of an immune response. So a fever shows that the vaccine is actually working, because that's what your body would do in a normal infectious state. It's important to talk to patients and parents about what they should expect following a vaccine so it doesn't take them by surprise. And there's a lot of people who will tell you to give paracetamol routinely pre-vaccination, post-vaccination. There is some evidence out there, that because paracetamol is affecting your fever and it is actually changing your immune response. So there is some research that suggests that routine use of paracetamol should be avoided because we don't know how it's going to change the immune response to the vaccine, and should only be used if a child reaches a fever at which you would normally treat with paracetamol. So prophylactic paracetamol use should be discouraged, mainly because we don't know what it can do. Also, no need to give a child a drug if they don't really need it. Uncommon adverse effects, and these are vaccine specific. Intussusception, which is basically where the bowel folds on itself, can occur following oral rotavirus vaccine in small children. There has been Guillain-Barré syndrome associated with influenza vaccine. Febrile convulsions can occur following any vaccine, particularly in children under 2, and children who are prone to that should be dosed in a specific vaccination clinic in order to provide appropriate support. Fainting. Fainting goes up in a lot of vaccines, usually in ones given to adolescents. It was particularly common during HPV bulk vaccination public health campaigns, particularly teenage girls at school are prone to fainting post-vaccination. So adolescents is definitely our key group to be looking at that for. Patients or parents of someone who's just been vaccinated should stay close by to a health professional for about 15 minutes, which will give enough time for any anaphylaxis or serious adverse events to present themselves. Nothing worse than walking away too soon and having something go wrong. So it's about 15 minutes that somebody needs to be observed following vaccination. Anaphylaxis is rare, but is the most serious adverse effect associated with vaccinations. Adrenaline must be available at any site that's providing vaccinations, whether that's nurse, pharmacist, doctor-led clinics. And the advice is always to over-treat what you think might be anaphylaxis than to miss anaphylaxis itself. So if in doubt, use the EpiPen or the adrenaline that's available, and always call 000 if you think anyone's having an anaphylactic event. Anaphylaxis to a vaccine does not prevent you having further vaccines. So it's about identifying it correctly and then dealing with it in the most appropriate way. So this is the Australian Prescriber Wall Chart. We got updated about a month ago. I've missed that, sorry. The doses are all still the same, but anywhere that provides vaccination should have access to this as a dosing information and a guide to observations and treatment of anaphylaxis. So other reactions that have been reported to vaccines. Now this is my favourite. Asthma, autism, particularly with MNR, diabetes from Hib, that's type 1, inflammatory bowel disease associated with the MMR, and that was linked with the autism by the same author, multiple sclerosis with Hep B, SIDS with all of them, but The science would say that there is no association between immunisation and any of these adverse events. Yeah, so vaccination should be considered safe and effective, and there is no link between all of these reactions. We have a very important way of reporting reactions using ADRACT. They can be reported to the public health unit that might be doing any of the vaccinations, and there is an adverse medicines event line for consumers. Any reaction we think to any vaccine should be reported in order for appropriate data. And it's particularly important that we have robust data for adverse effects related to vaccines in order to maintain community confidence and support of vaccination programs. So contraindications to vaccines. There are only 2 absolute contraindications to all vaccines. Anaphylaxis with that particular vaccine, or anaphylaxis with any component of that particular vaccine. There are a couple of contraindications for live vaccines, which is immunosuppression, and live vaccines should not be administered in pregnancy, and we don't recommend that women become pregnant within 4 weeks of receiving a live vaccine, particularly rubella. That's the one we're really concerned with there. But in the end, these are the only real contraindications to giving a vaccination. So different... Did I skip one? No, sorry. Ingredients in vaccines. There's lots of different ingredients used in vaccines for lots of different reasons. They have preservatives in order to maintain shelf life and allow them to be transported across the world. Vaccine products are used everywhere. And actually where they are most important and most needed is often the hardest place to get to. Adjuvants are in vaccines in order to present the antigen to the immune system to get the most appropriate and effective immune response, including aluminium. There are trace ingredients from the manufacturing process. So egg is the one that causes the most problems, particularly in flu vaccines because they're grown in egg cultures. Other things that can be present in vaccines in trace amount include antibiotics in order to prevent bacterial superinfections of the cell cultures, other chemicals, formaldehyde, glutaaldehyde, anything that might be used in that, so in trace amounts. And they're all clearly listed in the vaccine inserts if someone has an anaphylactic event to any of those things. So it's always important to double check if someone says they're allergic to vaccines, get a little bit more information and see if we can work through that and provide something safely. So mercury. Everyone's heard about mercury in vaccines. This is another pretty common question that I get asked. I don't want to give my child anything with mercury in it. Thimerosal was used commonly as a preservative in vaccines up until about the year 2000. It is ethyl mercury, not methyl mercury, which is associated with toxicity. Ethyl mercury itself doesn't accumulate in the body, whereas methylmercury does, and that's what causes the type of mercury that induces the brain damage and deliriums. So it's a completely different type of mercury. It's also different to the mercury that we find in fish. So if you eat tuna, you're already exposed to high levels of mercury from the environment. Ethyl mercury is different because it would be cleared in the fish as well. There is long-term evidence that thimerosal is completely safe and doesn't cause adverse effects, but we took it out anyway in order to keep consumer confidence in the vaccine programs and the safety of vaccines for their children. So it has not been used in any vaccines that are part of the National Immunisation Program since the year 2000 in Australia. It is in some Australian vaccines. It was in the Panvax influenza, and that was mainly because in order to get that product to market quickly, they had to put it out in a multi-dose vial as opposed to a single-use syringe. And that was about having enough preservatives in there to make sure it was stable and it was safe. So a risk-benefit assessment there. But you can confidently say that anything that's part of the Paediatric or National Immunisation Program does not have mercury in it in Australia today. So anaphylaxis can occur to the components of vaccines. Yeast, because HPV is grown in a yeast culture, so if someone's allergic to that, they wouldn't be able to have a HPV vaccine. Egg allergy, influenza, Q fever, yellow fever, and one particular brand of rabies vaccine are all grown in eggs, and that's how we get enough virus to get the antigens, so they can't be given to anyone with an egg allergy.
I thought that with influenza, like it's changed so much from when it is grown in the egg and things, or like that it's actually not a direct contraindication.
It's not. It depends on the level of allergy. So if someone was immediately anaphylactic to eggs, you'd still avoid the influenza. Yeah. But more and more brands are moving away from the eggs. It's actually a really expensive way to grow cell culture. So people are switching to the more labby way of doing it. The eggs are an old-fashioned thing that's sort of hung around, but yeah, more and more people are moving away from the eggs themselves. So that would be less and less of a problem. And it's about looking at like if someone gets a rash from an egg, then you would be comfortable doing that. So it's about looking at the anaphylactic responses and the risks associated with those. And they can still be given, so we just do it in a hospital or in an environment where there's enough supervision to make sure nothing goes wrong. So there are some additional precautions and risk factors you have to think about with vaccination. So doing pre-vaccination screening, detailed history to highlight any possible issues. For example, we had a patient present down to our special vaccination clinic in Peds late last year for his rotavirus vaccine, and it turns out he'd just been in hospital for intussusception, which can occur independently of the rotavirus vaccine. So making sure you've got an up-to-date, accurate medical history to make sure we're making the right decisions. You can delay vaccinations around periods of contraindication. You know, someone's pregnant, we can delay their vaccine until after the baby's born. We can change products, we can give a different schedule, we might recommend a different vaccine over another. There are things we can do to make sure people have what they need. And this is going to be particularly important for people who may need to travel, who require documented evidence of a vaccine in order to enter or leave a country. So false contraindications to vaccinations. All of these things. So a mild illness without fever, just if you've got a cough or cold, you can still have your vaccine. Family history of adverse events, past history of convulsions, you're on antibiotics, you can still have a vaccine. Inhaled corticosteroids, you hear this one. Any corticosteroids, oh, my child has asthma, eczema, hay fever, bit of a sniffle. Previous illness with anything that might be vaccinated for. Vaccination should not be postponed if someone's born premature, so we vaccinate based on your days from birth, not your days from when you were due. So everybody who's premature gets their vaccines exactly on time. It's particularly important. Jaundice, skinny kids, any neurological condition, infectious disease, a child's mother is pregnant, vaccinated breastfeeding, women to be vaccinated is breastfeeding, except for yellow fever. That's not a contraindication. Breastfeeding is not a contraindication. Recent or imminent surgery, often a really good reason to get vaccinated if you're coming into the hospital, definitely better. Or poorly documented vaccination history. We can take a blood test and work out what someone's been vaccinated for and is immune against, and design a plan from there. So very few of the concerns that people have about vaccination are actual contraindications to moving forward with vaccinations if they're open to it. And it's about engaging them in the conversation. So storage and supply of vaccines. Storage conditions are very important, particularly for live vaccines. Those bacteria and viruses are very fragile, and about keeping them in their optimum state in order to get the optimum response from your vaccine itself. Cold chain is particularly important from manufacturer all the way through to administration, and there's a national vaccine storage guideline called Strive for 5 degrees. All vaccines should be stored in monitored fridges, and it was always the pharmaceutical product we cared most about in fridges. We were community pharmacy, hospital pharmacy, we've always been very concerned about vaccine storage. And that's because if we're going to give someone an injection, we want it to work. Vaccines are often stored at home, so patients will get their vaccine from a community pharmacy and have to bring it to a healthcare provider. It's important they're provided with the correct advice about storage of vaccine in their home fridge if they have to do that. They shouldn't be stored on a door, they should be stored in the middle of the fridge, and there should be good circulating airflow around a vaccine to ensure that it's kept at the right temperature. The risk of storing a vaccine inappropriately means it may in fact be ineffective, and you will think you've been vaccinated against something and in fact you have not been. So reasonably serious consequences of inappropriate storage. So the Immunise Australia program is the federal government national immunisation program, which had its last big upgrade in 2013. It gets little upgrades here and there and little changes. I think the next big one will come within the next 12 to 18 months, and it will be the addition of the meningococcal B onto the schedule. Fingers crossed we get the funding for that. So these are all free supply vaccines, free catch-up vaccines. They're vaccine programs that go out into the community in order to get the most people vaccinated as we possibly can, and trying to reduce the incidence of these serious diseases in our society. And you can see from this picture what difference the introduction of a vaccine makes to the amount of a disease circulating in the community. So you can see here, as soon as we got the polio vaccine, completely went away. And I think if you asked your parents or grandparents and they remember what it was like in the 50s, everybody was pretty terrified of catching polio. People were in iron lungs, it was not a very pleasant disease. So Being able to remove that from the community is really important, but it's also about having a community memory of what it was like before the vaccines were here as well. And I think these days a lot of people don't remember what it was like to be frightened to swim in the public pool or play with your friends because you might get polio. Vaccine records are also important. Blue book that we use for babies. Red card, which we use for healthcare workers. We're not quite as strict here in the ACT. That's a New South Wales vaccination card. Travel requirements, so yellow fever. If you're travelling to a country that requires evidence of yellow fever, you need to have this particular yellow booklet that fits into your passport, so you can show people at border crossings that you've had your vaccinations as required. There's Australian Child Immunisation Register, so if you have a child who receives a vaccine, it gets uploaded into this register, so that data is available to fall back on. The similar register was set up for the HPV vaccination program register and also for Q fever. Q fever is a fairly Australian specific infectious disease, and the immunisation program extends to farmers as opposed to the general community, and it was about stopping that transmission in that particular group and seeing the vaccine was developed in Australia, so that register is about assessing its effectiveness and its ongoing development and use across the country. So the future of vaccines. Vaccines are not going anywhere. They're a very important part of our public health initiatives across the world. What people don't like about vaccines is the injection part. So people are very keen to develop new forms of vaccination that don't require large needles. Intranasal being the most popular choice there. So there's a lot of work going into particularly flu vaccines that can be given intranasal, and there was one on the market this year in Australia. So it was just a quick dose up, like a nasal spray up the nose, and it showed to produce a good immune response. So the nose is obviously highly vascular, so you can get the antigen into the bloodstream and get the immune response happening. Patches have been less effective. I'm not aware of any that have come to market yet, but people are very keen, as you can see in the picture, about popping the patch on the skin, which has little micro needles to get it in, and you just kind of pop it on and leave it and then pull it off in a couple of days. Haven't been as successful from a pharmaceutical development point of view. There's always hope to develop more vaccines for other diseases. HIV, keeps coming up. Cancer, so melanoma, particular stimulation of the immune response against melanoma cells when they're first presenting, as opposed to by the time they're visible to the naked eye. And dengue fever is another popular one. Dengue causes not a lot of mortality, but a lot of morbidity in communities where dengue is endemic, and a lot of lost work days and costs to the community from that. Ebola recently came back up in the news, which excites me because I'm a nerd. The WHO regards Ebola vaccines as a high priority, and that's because the transmission rates of Ebola, it just spreads so quickly, and the vaccine is the most effective way of stopping that. So 2 vaccines had clinical grade trials available in the last Ebola outbreak in Liberia, and they've just used those again in the new one that happened about 6 weeks ago. They brought those vaccines out again and vaccinated people, and it stopped the outbreak of Ebola very quickly. Did anyone even know there was an Ebola outbreak around ago? Didn't blow up like they can tend to, because they got the vaccine to them quickly and are doing more clinical trials in that space. These trials have been accelerated very rapidly. Usually it takes a decade to bring a vaccine to market with appropriate studies and testing and development. So there's always the ethical issues about these people are actively in a critical situation where they might be exposed to a disease. Is it ethical and reasonable to test a vaccine on them? Hard to say, really. I mean, I think the World Health Organization has moved towards using the vaccines and trying to do what they can because it's better than nothing, but there are always going to be some issues associated with that. Ebola also, they used a lot of passive immunisation to stop the last big outbreak, using serum from recovered patients to get the antibodies and purify those and re-administer them to people who were sick to get them better quickly. And supply issues. These outbreaks, same as the polio outbreaks that are still currently occurring, they're in places that are hard to reach. They're places that don't have good infrastructure, not good roads, not good running water, not good refrigeration, and they're in places that are affected by war and terrorism. So it is hard to get a vaccine to these places, and maintaining supply once you start to make sure it's an equitable distribution across the community. Community concerns keep coming up about vaccination. There's a lot of really vocal anti-vaccine groups around Australia and the world. They have a lot of safety issues about vaccinations, and I hope I've addressed some of the common questions and concerns that people have throughout this session. They're concerned about the excipients, the aluminium, the mercury, all these things in vaccines, the overloading of the immune system. So vaccines cause autism, which has been disproved in many large clinical trials and databases. That it's not a natural form of immunity, and it's better to get the infection itself so you've got a natural immune response as opposed to the artificial. And other generalised concerns about, inflammatory bowel disease and diabetes and things like that. So community concerns are important, and they need to be addressed in a way that is supportive and engaging, and it's about engaging people in the discussion about vaccines and why they're important. So an example of the global effect of the anti-vaccination movement. So Europe, in the UK, particularly Wales, there have been more than 8,000 cases of measles since 2012, and that 82% of those people were unvaccinated. So because of low vaccination rates in the community, measles has spread. Poland has a lot of rubella cases, so 21,000, where the vaccination status was known, 89% were unvaccinated. Rubella having particular concerns because rubella infection in pregnant women causes a significant amount of developmental delay and blindness in babies who are exposed to rubella in utero. Rubella through Japan, and measles in New York City. So this was a particularly interesting outbreak because it was a high socioeconomic area of New York where someone hasn't vaccinated their child who was exposed to measles on a trip to Mexico and brought it back and shared it with all his playgroup mates. So unfortunately some of those children weren't able to be vaccinated because they were too young, or some of them had not been vaccinated due to parental choice. So 100% of the cases in that 34 kids at daycare, 100% of the kids who got measles were unvaccinated. Not really very fair on the kids who were too little, who really had no other option other than herd immunity protecting them from that. So the anti-vaccination movement has had global effects on a large scale. We've seen cases of diphtheria in Australia, which is the first time in 50 odd years. We've seen a lot of cases of rubella across the world, and measles has become a common infectious disease again in a lot of places. And with the speed of air travel and the amount of travelling people do, it's spreading very quickly. And we do have cases around Australia from time to time. Pharmacists, we do a lot of supply stuff, but we can also provide evidence-based advice and reassurance and answer questions people may have about vaccines, support parents in decision-making processes, answer questions with evidence and fairness, acknowledge the side effects, because side effects do exist to vaccines, but then talk about the benefits, and make sure people are aware of the risks of not vaccinating, or vaccinating according to their own delayed schedule. There's lots of places you can call for immunisation around Australia. Paediatric Immunisation Clinic, ACT Medicines Information, and the Immunise Australia hotline. And the Immunise Australia handbook is available in full online through health.gov.au. So, yeah. Come back next week. Hear more about vaccines.
Thanks.